In this video, we will discuss about this cathepsin K protease efficacy in the vicinity of the O-glycosylation site and its specificity towards mucin. So far, the researchers have tested this cathepsin K protease activity on the purified mucin and also on the different kind of cell lines which express this mucin. And in both the cases, they are able to see the activity of this cathepsin K in the form of degrading this mucin length. So now they wanted to move further and wanted to check whether the cathepsin K can cause cleavage at the near the O-glycosylation site and whether it is specific to the mucin or not. So for that, the reason why they decided to check this thing, because the activity of the trypsin get many times hindered by this glycocalyx or the glycosylations through these glycosylations near the cleavage site that prevents the accessibility of the cleavage site by the proteases such as trypsin. So here, in order to check this hypothesis, they use the motifs of the O-glycopeptides, that is from the residue 24 to 47 that can be cleaved by CTSK, and simultaneously for checking the specificity they used motif from non-modified peptide, and there is a third motif which contains both the things. And thereafter what they did is they treat them with this CTSK, and afterwards they did the glycoproteomics on the peptide, and here they have represented the sites through the glycan symbols, and this does not represents the diversity of the glycans at the particular site, but it only depicts the number of sites at the number of glycosylation sites within these residues, and the bar plot represents the number of cleavage or the peptide cleavage that they are able to find out at a particular site. So in this case, what they are able to find out, that in the case of this motif from O-glycopeptide, they are able to cleave near the site of this O-glycosylation. It can be seen here in most of the cases, and these things they have validated using this MYZ analysis. Now, on the other hand, or we can see that surprisingly, they are also able to find out the peptide cleavage at the O-glycosylation site even in this motif from non-modified peptide, and for this case they use residues of this PSGL1 from 148 to 197, and for the previous case they use MUC1 residue from 24 to 47. So in these PSGL1 residues they are able to see the peptide cleavage at the site of the glycosylations that can be seen here, and based on the previous publications that although it does not require any specific sequence, but it prefers proline and threonine compared to the other amino acids in the vicinity of the cleavage site. And this thing, for example, in this case the proline is there nearby to the site, and even in this case the threonine, it cleaves at the threonine sites. Similarly, for this MUC1 residues also they saw the similar pattern, that the cleavage is at near the proline site and near the threonine sites. So, and all these are in the vicinity of the O-glycosylations. And the reason why they selected O-glycosylations, because most of the cancer cells are this covered by the heavy O-glycosylations, and that prevents heavy O-glycosylations near the cleavage site. That prevents, and most of the time prevents, the proteases activity. For example, the trypsin's activity gets affected because of the heavy glycosylations near its cleavage site, and this results for the PSGL residues 148 to 197 are also validated using the mass spec, indicating that this uh, peptide cleavage of the mass spec, since mass spec is uh, right now the gold standard to validate the molecules or the peptides or the glycans. So from this they are able to come to a point that this cathepsin K protease efficacy works in the vicinity of the O-glycosylation sites that we have seen, and it also has it is not only specific to the mucin, as we can see the cleavage or the protease activity even in these PSGL1 residues. So this is how they validate their hypothesis. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.